Hi, everyone, and welcome to our webinar. My name is Kasia Proctor, and I'm the Director of Marketing Communications here at Biolog. I will be your moderator today. Today's webinar is titled Microbial Community Analysis Made Easy with Odin Software. First, I have a few housekeeping items. We are going to send the recording of the webinar to everyone who had registered. If you have any questions, please submit them through a Q&A chat feature. And that's it. Now let me introduce our speaker. Max Gravener is our product manager for all things Odin, instrument and software. He got his PhD from University of Georgia in microbiology with a focus on fungal genetics, where he worked with the Candida albicans, studying natural variations among clinical isolates and how gene expression and regulatory differences can impact their ability to form biofilms on implanted medical devices. Welcome, Max. Take it away.
Thanks for the introduction, Kasia. I'm really excited to talk to you all today about the great tools included in the Odin Community Analysis software package and demonstrate how they can be of help in a variety of ways for your research. Microbes don't live in isolation, as you may know. They form complex communities where each member carves out a niche, consuming resources and producing byproducts, which can be used by other members of the community or by plants and animals. Studying these communities is a challenge surmountable only through the utilization of a range of analytical techniques to identify key features of the community and how those features change in response to community structure and environment. A variety of omics techniques are used to study any microbiome, from genomic methods like NGS and 16S to transcriptomic methods studying gene expression, proteomics and metabolomics, which rely largely on chromatography and mass spectrometry. As we travel up in the chain of various omics, there are more regulatory mechanisms, meaning a change in the genes might not necessarily correlate with phenotype. Fortunately, at the end of this chain is phenomics and the metabolic phenotyping, which serves as the output of all of the regulatory mechanisms and what biolog assays measure. So where does the Odin Community Analysis Toolkit come in, and what does it look like? Well, first comes the Odin instrument, obviously, and the community analysis plates, which do all of the heavy lifting for incubating, reading, and challenging the microbial communities in a variety of conditions and substrate utilization tests. The other half of the toolkit is the Odin analysis software, which comes with powerful analytical metrics that we'll talk about shortly, each of which have their own unique benefits. Together, this system gives you the ability to really gain a good understanding of your microbial communities and take action based on your results, such as adjusting the structure of a community to tune its phenotypic output. Before we dive into the software, I wanted to go over a little bit of background on Biolog microplates and Odin. Biolog microplates generally contain a variety of metabolic substrates like carbon or nitrogen, or inhibitory compounds like antibiotics or salt, where depending on the plate type, each well is a unique condition, or the plate may contain replicates for additional statistical power. Some plates also contain a redox dye, which is reduced to a purple color in response to energy production, which gives you the flexibility to measure growth or metabolic output over time. The Odin instrument serves as a platform for incubating and kinetically reading up to 50 plates at a time in the case of Odin L, or up to 8 plates with Odin 8. Both Odin instruments use optical density to measure both growth and metabolic output, where it can be visualized in real time in the software and be used to both characterize single isolates and communities or identify thousands of different microbes. Using Odin for kinetic phenotypic characterization is highly complementary with genetic profiling, making it an ideal tool for screening large libraries and complex communities, which may have different phenotypes depending on their environment, or identify key functional changes in mutants. Screening across a large library of conditions can also be helpful in optimizing culture conditions to boost production in industrial strains. So why bother with kinetic data? Well, some phenotypes manifest only as differences in growth over time, like in this example where culture B took longer to begin metabolizing the substrate, but both cultures eventually reached the same endpoint. So how do we handle the data analysis? Well, that takes us into the Odin software. The Odin software is a purpose-built platform driven by user feedback, which includes a variety of analytical methods. With the Odin software, you can really get a great understanding of your microbes. For instance, you can create and read up to 50 plates simultaneously, meaning that many projects can be running at once. You can get statistically significant results comparing samples' growth curves and generate scatter plots. You can screen your community's metabolic and growth profiles, determine minimum inhibitory concentration, rank rhizosphere microbes for phosphate solubilization and nitrogen fixation, and identify over 3,000 bacteria and fungi, just to name a few features. While there's a lot of exciting possibilities with the Odin software, today we're going to be focusing on just community analysis. The first community analysis metrics that we're going to be talking about today are average well development and Shannon's indices, which are really helpful in determining the overall metabolic health and flexibility of your community. Before we get into the specifics of how these metrics are calculated, let's first talk a bit about why you would want to use them in the first place. Consider this. You have a complex microbiome sample from the environment with an unknown composition, and you want to get a picture of its overall metabolic flexibility and preferences without having to go through all of the effort of isolating every single strain in that community. Well, with average well development and Shannon's index tools, it's really very easy to get a holistic view of the community flexibility and activity from multiple angles. This saves you the time and effort it would take to separate out all of the constituent microbes without the risk of losing any community-level phenotypes that would otherwise be really easy to miss. Now let's get into the gritty details of how these metrics work. The average well development, as the name implies, takes the average of either growth or color development across the entire plate, which shows the overall metabolic output or growth over a variety of substrates. And this can indicate the overall metabolic output of the community. Shannon's diversity index is related to the number of substrates on the plate which are utilized, where a higher diversity means that more substrates are in use. This can give you useful insights into the flexibility of your community. Shannon's evenness is related to how much of each of these substrates are utilized, where a high evenness index means there's uneven utilization, and an index of 1 means that all substrates are used evenly or that all substrates have reached saturation. This is useful in showing that while a large number of substrates may be used, utilization may be skewed to only a few. Together, these three metrics allow you to compare complex communities and get actionable insights without necessarily knowing what those communities are made of. Let's take a look now at a real-world example of how these metrics can be used to great effect by characterizing a community taken from the popular fermented beverage kombucha. To do this, we used our PreBioM plates. The PreBioM plate line is a set of 3 microbial phenotyping microplates packed with prebiotic substrates. The phenotyping plates cover a menu of prebiotic substrates, increasing in complexity from simple sugars to macromolecules. And these can be used independently or as a set, and we're going to use all 3 here. So PreBioM 1 contains a mix of mono and disaccharides, PreBioM 2 contains more complex poly and oligosaccharides, and PreBioM 3 has a variety of fibers and food extracts. The plates take advantage of built-in triplicates as pictured in the plate, enabling statistical analysis in the Odin software. Kombucha is a fermented tea beverage pioneered in China, which has been made for centuries simply by taking advantage of the vibrant microbial community present in tea leaves. The SCOBY, or symbiotic community of bacteria and yeast, is housed within a cellulose biofilm that can support dozens of different microbes in a metabolic cascade, processing both the natural tannins and other components of tea, as well as the sugar used to sweeten the tea, where each microbe has a responsibility to process a specific substrate into something else that can be used by other microbes in the community. Here we brewed tea and fermented it into kombucha for 2 weeks, and then took some samples. The microbes were then separated from the tea, and the entire community was then inoculated onto PreBioM plates 1, 2, and 3, along with a redox dye, before being placed into Odin for incubation and reading to learn more about their carbon source utilization preferences. We hypothesized here that the community would be fairly robust given its variety and structure, and that it would likely process a wide variety of prebiotic substrates. So let's take a look first at the substrate level utilization before getting into the overall growth patterns and trends with the community analysis tools. So first, taking a look at prebio M1 or mono and disaccharides, we can see that unsurprisingly, there are a lot of simple sugars which are utilized by the community. This makes a lot of sense given that many of these substrates like sucrose, fructose, glucose, and others don't really take that many steps to process and are likely being broken down by many different microbes within the community. So let's take a look now at the more complex substrates in prebio M2 and 3. And here we can see that not as much energy is produced on any given poly and oligosaccharide here on prebiome 2 in the middle. However, there are many plant-derived fibers and extracts, like those on prebiome 3, that are being utilized by the community. So this could be due to the pressure on that community in the kombucha to utilize substrates that are derived from the tea leaves. So let's move on to the average well color development and Shannon's indices to see if any other trends start to emerge. Using prebiome M1 as an example, if we take a look at each of these curves independently, we start to see some trends emerge. So first, looking at average well color development, we can see that energy production picks up at around 20 hours before starting to even out. This is further reflected by the jump in Shannon's diversity, which means that several substrates are all being utilized. Pair that with the fact that the evenness index spikes and quickly comes down, we can see that while initial utilization is very uneven in a small number of substrates, the early use substrates quickly all reach the same level of energy output. If we took a look back at our substrate level kinetic curves, we can tell that this first push of energy production is largely driven by simple sugars like mannose, fructose, glucose, and much to my surprise, sucralose. And now this is where things start to get a bit more interesting. A second increase in metabolic activity begins to arise around 60 hours of incubation, according to the average well color development. And we can see that diversity begins to rise as well, meaning that more substrates are being used, perhaps indicating that to switch metabolic programs over to process them takes some more time, or that these microbes that are responsible for that processing are slow growing. Interestingly, the evenness does not spike, but rather shows a modest gradual increase before slowly coming back down, meaning that these late blooming substrates are more evenly utilized than that first wave. If we go back to our substrate level curves, we can see that sucrose, the primary sugar in kombucha, here in the bottom left, is one of the driving forces of this late phase growth, along with other sugars like arabinose, turinose, ribose, and maltose. Taken together, the community analysis metrics, along with substrate level curves, clearly define a biphasic growth pattern linked to substrate type. Now that I've shown you a real-world example of how this data can be used, let's go into the software now to take a look at how these curves are generated.
Here we're going to be taking a look at two microbial communities from different soil samples. So first I'm going to pick out the community analysis project I want to analyze. And then for a reference sample, I'm going to pick CM1, which comes from soil rich in decaying organic matter. The test sample I'm going to compare to CM2 comes from dry compacted soil with little organic matter. I'll then select our plate type and hit generate charts to get things going. So taking a look first at average well color development, which, as the name implies, takes an average of the signal in all wells over time and enables you to get a bird's eye view of the overall energy output of your community. Unsurprisingly, CM1, shown here in black, shows much more robust energy output than CM2, shown in orange, which is consistent with having many microbes working actively to break down organic material. Shannon's diversity index here refers to the diversity of substrates which were utilized by the community. A high diversity index means that many substrates are being used. Comparing CM1 and CM2, we see that CM1 shows a rapid increase in diversity of substrates early on in incubation, whereas CM2 lags behind and does not metabolize the same variety of substrates, as indicated by the fact that it does not reach the same height of Shannon's diversity index. Noise caused by high background in this index can be controlled here using the pattern development cutoff, which is the total signal in all positive wells. Shannon's evenness index here is related to the rate at which substrates are utilized. As evenness approaches 1, substrate utilization has reached an equilibrium, where a spike can mean that only a handful of substrates are being used. Finally, CM1 shows a steep spike indicating uneven use of a few substrates before slowly coming down to 1 as more and more substrates are utilized. CM2, only using a few substrates, shows a faster drop to equilibrium. Noise caused by high background can be controlled here using the positive response cutoff, where only wells which reach that threshold of absorbance can be counted.
Moving on to our next and newest community analysis tool, functional guild analysis, which can be instrumental in identifying problem pathways within your community. Ecoplates containing 31 carbon substrates are a powerful tool that are widely recognized within the field with hundreds of publications that are great for carbon source preference screening for microbial isolates in communities. Ecoplates are a great screening platform for carbon source utilization as they're able to accept a wide range of sample types such as pond water, soil suspensions, purified microbes, and many, many more. The ecoplate contains 31 pre-plated substrates, which are listed out here, which were selected to probe a variety of pathways known to be important to environmental samples. While individual substrate utilization patterns are informative, it can be equally important to assess trends among substrates of the same type, otherwise known as functional guilds. Carbon sources on the ecoplate can be divided into 5 functional guilds. We have carbohydrates, amino acids, carboxylic acids, polymers, and amines. Substrates within each of these groups require shared mechanisms or pathways to process, so it can be useful to compare the guilds to one another to more deeply understand the metabolic profile of any given isolate or community. Functional guilds are a great way to pinpoint key pathways that are important for a given community or isolate. Communities, like those found in soil, can be incredibly diverse with a broad range of activity and substrate preferences. On the other hand, small or inactive communities can have trouble processing certain types of substrates. Whether a community is active or inactive can be greatly influenced by where that community is, what it's composed of, or the nutrients that are available to it. When it comes to communities, the more vibrant and active, the better, so that it can handle anything that you throw at it. Here we have an example of 2 different communities from different soil types, which show drastically different energy outputs. CM1 shows relatively high activity in each of the 5 functional guilds, meaning that it has a higher overall flexibility. Meanwhile, CM2 struggles to produce energy using anything except for basic carbohydrates. If this was your soil, this would indicate potential problems with nutrient turnover and might point to a need for adjustment, whether that be through adding microbes to the community or increasing the availability of preferred substrates. Now that we've seen an example of how functional guilds can be used to compare samples, let's take a look at the software to see how to make that happen.
Here we're going to be taking a look at 2 microbial communities in different soil samples. So first I'm going to select the project where my data is stored, and then I'll pick a reference sample. In this case we're going to look at CM1, which comes from soil rich in decaying organic matter. The test sample I'm going to compare to is CM2, and this comes from dry compacted soil with little organic matter. We'll then pick out our plate type, and then select area under the curve for our parameter, and then hit generate charts to get things going. Taking a look at the average area under the curve, or total energy output over time for each functional guild, it's really clear to see that the community CM1, shown here in orange, has a much more robust metabolic network, generating significantly more energy overall compared to CM2. This makes sense given that CM1 should be a more diverse and active microbial community as it came from soil with lots of actively decaying organic matter. Additionally, we can see that while CM1 and CM2 are nearly evenly matched in terms of energy produced with carbohydrates, the differences are more evident when it comes to the processing of more complex substrates like amino acids, polymers, carboxylic acids, and amines. This indicates that there may be a lack of microbes in the community for CM2 which are able to break down those substrates, or that the pathways required to process those substrates are absent or being turned off.
Let's move on now to using the Community Analysis Toolkit to quantitatively rank rhizosphere microbes in their ability to solubilize inorganic phosphate and fix nitrogen, which are two key functions of the rhizosphere and are required for plants to flourish. First, let's talk about nitrogen fixation and how it's typically measured in microbes. So, typically, people use the acetylene reduction assay, which is a highly sensitive assay where acetylene gas is used as a proxy instead of nitrogen gas, where microbes will reduce the acetylene into ethylene, and then the technician will use a gas chromatography machine to measure that conversion rate. However, this doesn't actually directly quantify nitrogen fixation, it just uses a similar pathway. There are a couple of downsides to this methodology. First off, it's really labor intensive and time consuming. It has a limited dynamic range, and it also uses an explosive gas in sealed containers. Now let's talk about phosphate solubilization. The classic PVK assay uses inorganic insoluble phosphates suspended in agar plates, and it takes microbes plated on a disc and looks for zones of clearing around that disc, which is produced whenever the microbe secretes an acid and solubilizes that phosphate. The problem with this method is that it's difficult to make these plates consistently, and it's only qualitative. There's no quantitative measurement going on here. So how do we address the deficiencies of the traditional acetylene reduction assay and PVK assays for quantifying and measuring phosphate solubilization and nitrogen fixation? Enter the rhizoplates. Rhizoplates are an easy-to-use solution for quantitatively ranking microbial candidates or communities on their relative ability to solubilize inorganic phosphate in the case of rhizoplate P, or their ability to fix atmospheric nitrogen, as with rhizoplate N. Rhizoplate N is a great alternative to the acetylene reduction assay, which requires a high degree of training and the use of that GC mass spectrometer in dangerous acetylene gas. Rhizoplate P, meanwhile, replaces the qualitative PVK assay, which relies on difficult-to-make agar plates and measuring zones of clearing. Both of these assays give invaluable insight into the agricultural utility of candidates and communities, and also provide useful information about carbon source utilization, as each plate contains 30 different carbon substrates, which can impact nitrogen fixation and phosphate solubilization. Let's take a look now at a real-world example where candidate engineered strains are being ranked for their nitrogen fixing ability by plotting their normalized average well growth development and calculating the area under the curve for each of those plots. This gives us a quantitative way to rank the candidates. So here we used a couple of positive control strains commonly found in the rhizosphere, A. brassilens and D. gamosa, which are both known to fix nitrogen. We also included two strains which are known to not fix nitrogen as our negative controls, S. epidermidis and E. coli. Finally, our candidate engineered strains, C1, C2, C3, and C4, which are candidates for adding to a soil. So we use the Community Analysis Toolkit to plot the normalized average well growth development for each of these samples. We can clearly see that C1 and A. brassilens, here shown in black and cyan, are the top performers. Then to give us a quantitative metric, we also tabulate the area under the curve for each average well growth development curve to get a full ranking of the samples. The top two strains are distinguished here based on their average well growth development area under the curve values, and although C1 shows the highest average well growth development at the endpoint, A. brassilens slightly outperformed it. Additionally, with our negative controls showing near zero values, we observed that two other strains, C2 and C3, also demonstrated significant nitrogen fixation. Now let's take a look at phosphate solubilization on rhizoplate P. We again used two rhizosphere positive controls, but this time A. piti and P. petita, known to solubilize inorganic phosphate, and we used those same negative controls in test strains as well. When we plot the average well growth development, we can see that A. piti was the top performer, here shown in black, followed by candidate C3 and P. petita in third. However, when we take into consideration the area under the curve of each of these plots, we can see that the true ranking shows both positive control strains outperforming C3, although it's not far behind. Even for P. petita and C3, which intercept at 75 hours and share similar endpoints, we can make a clear determination on rank using the area under the curve. These nitrogen-fixing strains are clearly separated from the negative controls like S. epidermidis and E. coli. However, this is only part of the story. With the full data in hand, we can now see that one strain, C3, predicted to exhibit both nitrogen fixation and phosphate solubilization, performed exceptionally well in both assays, and would therefore be a strong candidate for a biostimulant product. Rhizoplates in Odin are a really great solution for replacing the acetylene reduction and PPK assays, which have some significant issues as we've discussed. As shown in the example, kinetic monitoring, not just endpoint reads, are really critical in determining a quantitative ranking of these candidates and controls. It was clear to see that with a combination of average well growth development plots and calculated area under the curves, that the C3 candidate was the best of both worlds with nitrogen fixation and phosphate solubilization. This method can not only be applied to purified isolates, but also to communities by leveraging the wide array of carbon substrates available in the rhizoplates, meaning that you have more power to develop sustainable and improved biostimulants. Now that I've shown you how we can apply the rhizoplate average well growth development normalization and analysis, I want to get into the software to show you how to actually generate these curves.
So first, I'm going to select the project where my rhizoplate data is stored. And then I'm going to pick our reference sample. In this case, we're going to use E. coli, which is known to not solubilize inorganic phosphate. And then I'll pick out our test strains. In this case, we're going to be using A. piti and P. petita, which are bacteria commonly found in the rhizosphere. Then I'll pick our plate type, and we'll hit Generate Charts to get things going. If we scroll down here and take a look at our average well growth development curve, we can see that the signal here is really low, and that's to be expected as inorganic phosphate is the sole source of phosphorus for these microbes, and it makes it very difficult for them to grow. So let's adjust the y-axis maximum here in order to better view our data and then regenerate the charts. Next, we need to look for the latest point where the growth curves first level off and then pick up again, which indicates when they ran out of stored phosphate and start using the exogenous or inorganic phosphate for energy. So here that looks to be at about 25 hours with E. coli, our negative control. So if we enter 25 hours as our x-axis minimum and then regenerate the charts, the software will automatically normalize the data and to remove all growth signal prior to phosphate solubilization. And this allows us to rank our samples by which we're most effectively able to grow by solubilizing the inorganic phosphate. With our data normalized, it's really clear to see that A. piti, shown here in orange, was the most proficient at solubilizing the inorganic phosphate, followed by P. petita in green, whereas E. coli struggled to surpass the baseline, which is to be expected as it was our negative control. And finally, to give you a quantitative metric by which to compare these samples, you can also export the AWGD plot parameters area under the curve as a CSV file.
I hope that this presentation has convinced you of the power of the Odin community analysis software in gaining actionable insights which can lead to impactful products. When considering which part of the community analysis toolkit to use for your application, ask yourselves these questions. Do you need to compare communities to determine efficacy in nutrient turnover? Consider the average well development in Shannon's indices, which can show the overall health and flexibility of your microbial community or isolates. Do you need to find deficiencies in an existing community or monitor change over time? Use the functional guild analysis tool, which can pinpoint missing metabolic pathways or niches for you to target. Do you need to find the best candidate for improving soil health and therefore crop yield? Think about the rhizoplate average well growth development analysis to quickly and quantitatively rank your strains and communities. At the beginning of this talk, I mentioned that the Odin software is driven in no small part by feature requests from our users, and the new custom plate map feature is a perfect example of this. New with Odin 3.3, you can now create your very own custom plate types, which can be selected during batch creation, just like any other biolog plate. This means that you can create just the right plate for you and your needs. Not only can you create the plate, but you can also analyze it with the full power of the average well development, Shannon's indices, and functional guild tools, as well as many other features available through the phenotyping package for the Odin software. Speaking of phenotyping, don't forget to look into our other great tools at your disposal if you have the community analysis package already. I don't have time to cover everything, but you can calculate p-values, minimum inhibitory concentrations, and a host of other useful parameters, as well as generate publication-ready figures among many other useful features. Before I end, I just want to take a moment to talk about the 3 pillars of Biolog, products where we support your workflow. You've heard me talk a lot today about Odin and phenotypic and community analysis, but we also have our ID station for identification and our anaerobic chamber line. On the consumable side, we have a variety of microplates for you to use to characterize and identify plates, as well as a wide variety of anaerobic culture media. And then finally, in our lab services arm, we have a fully certified lab that can help you with identification, phenotyping, enumeration, and a myriad of other services. Thank you all very much for attending today's webinar. I want to invite all of you to ask any questions you might have in the chat, or if we don't get to it today, feel free to email us at info@biolog.com. At this point, I'll turn it back over to Kasia, who will direct us through our Q&A session. Take it away, Kasia. Thank you.